Now we discussed about the cell division. Today we discussed about mitosis. There are two types of cell division, mitosis and meiosis. Mitosis occur in all over the body except reproductive organ. Meiosis or reduction division took place in reproductive organs. But now we discussed about the different steps of mitosis. In case of mitosis and meiosis, cell division start with karyokinesis. That means the division of nucleus and followed by cytokinesis. The cell division with two parts. First, karyokinesis. Kinesis means division. Karyokinesis. And second phase, cytokinesis. Karyo means nucleus. First, nucleus is divided into two and then the cytoplasmic division occurs. First, we discussed about karyokinesis. That means the division of nucleus. Karyokinesis. Mainly there are four phases: prophase, metaphase, anaphase and telophase. The nuclear division with four phases. First, prophase. Second, metaphase. Third, anaphase. And fourth, telophase. First, prophase. Prophase is again divided into three: early prophase, middle prophase and late prophase. What about early prophase? Early prophase. Here we draw a picture of a typical cell with nucleus and the cytoplasmic part. The protoplasm means the cell content with nucleus. Without nucleus, it means cytoplasm. The protoplasm with the nucleus and cytoplasm to form protoplasm. Cytoplasm means the protoplasm without nucleus. Now here we can draw a simple picture of a cell. In case of early prophase, centriole. We know centriole is a cell organelle. There are different types of cell organelles. Centriole, that here we draw the animal cell. Centriole is a cell organelle, helps for cell division. Then nucleus, here we can draw nucleolus and chromatin reticulum. This is nucleus, chromatin reticulum, nucleolus, this is centriole, cell organelle. Centriole. In case of early prophase, the centriole divided into two and move towards the opposite poles. This centriole divided into two and enter into opposite parts. Chromosomes start coiling. This chromatin reticulum coil to form chromosomes. Chromosomes start coiling, nucleus enlarged and become less distinct, not clear. In case of early prophase, centriole divides into two and moving towards opposite poles. Chromosome start to coiling. The chromatin reticulum start to coiling to form chromosome. Chromosome start to coiling. Nucleus enlarged and become less distinct, not clear. Nucleus enlarges and become less distinct. Now complete the early prophase. Next, middle prophase. In middle prophase, chromosome condensation complete and the chromosome become shorter and thicker. The chromatin reticulum condensed to form chromosome. The chromosome again continue the condensation process and complete to become shorter and thicker chromosome. Now we draw the picture. Here we draw two chromosomes. In case of middle prophase, chromosome condensation complete. Chromosome condensation complete. Become shorter and thicker. The chromosome become shorter and thicker. Shorter and thicker. Each chromosome made up of two chromatids. Each chromosome made up of two chromatids held together at their centromere. This is the centromere. These are two chromatids. Each chromosome made up of two chromatids held together at their centromere portion. Held together at their centromere. Each chromatid contains newly replicated daughter DNA molecule. Each chromatid, these are two chromatids, each chromatid contains newly replicated daughter DNA molecule. The chromosome made up of DNA. Genes are the part of DNA strand. Each chromatids Each chromatid contains newly replicated daughter DNA molecule. Daughter DNA molecule. Chromosome is made up of DNA. Genes are the part of DNA strand. Next, the third part of prophase, that is late prophase. Late prophase. What about late prophase? Spindle fibers extend from poles of equator. Now, again, the nuclear membrane already diluted. The chromosomes are freely float in cytoplasm. In late prophase, spindle fibers, these are the spindle fibers, spindle fibers. Spindle fibers extend from poles of equator. Poles of equator. Nuclear membrane and nucleolus disappear. In late prophase, spindle fibers extend from poles of equator. These are the equator region. Then nuclear membrane and nucleolus disappear. That dissolved. Nuclear membrane and nucleolus disappear. Now complete the 3 steps of prophase. Next step of mitosis, that is metaphase. All chromosomes arrange in equator, that is central portion. Spindle fibers attach to centromere. All chromosomes arranged in equator, that means central portion. Equator, that means central portion. Spindle fibers attached to centromere. Spindle fibers attached to centromere. We can draw the picture of metaphase. The chromosomes arranged on equator region. These are the centromere portion. The spindle fibers attached to centromere. That arise from, the spindle fibers arise from centriole. The spindle fibers arise from centrioles and attached to the central portion of chromosome. These are centrioles. This is centromere. Centromere of chromosome. Centrioles are the cell organelles. Centromere is a joining part of two chromatids of the chromosome. Next, third phase, anaphase. Anaphase. Anaphase is the third phase of mitosis. Here, in case of anaphase, spindle fibers contract. Spindle fibers become contract and pull the centromere to opposite poles. Half number of chromosomes move towards opposite poles. The spindle fibers contract and pull the centromere to the opposite poles. Half number of chromosomes move towards opposite poles. Now we draw the picture. The last phase, telophase. Last phase of karyokinesis, that is telophase. Two daughter nuclei formed in opposite pole of the parental cell. Two daughter nuclei formed in opposite poles in the parental cell. Now we draw the picture. Two daughter nuclei formed in opposite poles. In the parental cell. This is the parental cell. These are the two newly formed nuclei. The nuclear membrane and nucleolus reappear in daughter nuclei. The nucleolus and nuclear membrane reappear in daughter nuclei. Nuclear membrane and nucleolus reappear in daughter nuclei. Now complete the karyokinesis, that is the division of nucleus. The karyokinesis followed by cytokinesis, that is the division of cytoplasm. That means cytokinesis. Kinesis means division. Here is some difference between case of plant cell and animal cell. First we discuss about the cytokinesis of plant cell. In plant cells, phragmoblastes or cell plates begin to form the center of parental cell and extend towards the periphery. Plant cell. This is a typical structure of plant cell with two daughter nuclei. Phragmoplastis, that means cell plate. Or cell plate. Phragmoplast or cell plate begin to form from the center. They begin to form at the center of parental cell and expand towards the periphery These phragmoplasts formed at the center portion and extend towards the periphery in case of plant cells. And the plant cell divided into two with the nucleus. In case of animal cells, this is animal cell. The cytoplasmic division, in case of cytoplasmic division or cytokinesis of animal cell, invagination, that is make a groove, invagination of plasma membrane proceeds from periphery of the cell towards the interior. In case of animal cells, invagination of plasma membrane proceeds from periphery to the cell towards the interior. The cytoplasm divided into two daughter cells forming two nucleus. Invagination of plasma membrane. The last portion, importance of mitosis. Importance of mitosis. What are the importance of mitosis? It is the only mode of reproduction in unicellular organism. It is the only mode of reproduction in unicellular organisms like amoeba, paramecium, euglena, etc. There are a lot of examples, but we discussed about some 2 or 3 examples of unicellular organisms. It is the only mode of reproduction in unicellular organism. It is a process by which growth takes place in multicellular plants and animals. But in case of multicellular animals, it helps for growth. The mitosis helps for growth of the body in case of multicellular animals. Then the mitosis play a role in repair during wound healing, regeneration, etc. In multicellular organism, helps for growth. Mitosis helps for or play role in repair during wound healing, Regeneration. What is regeneration? The formation of lost part is known as regeneration. In case of planaria, we can see complete regeneration. But in case of wall lizard, that is a partial regeneration of tail portion we can notice. Regeneration, etc. The unlimited mitosis causes the production of abnormally formed cells named cancer cell. That is not important. That is a bad scenario of mitosis. That means unlimited mitosis cause for cancer cells. Unlimited mitosis causes the production of abnormally formed cells. Immature cells, production of immature cells, abnormally formed cells named cancer cells. In case of plant cells, in case of plants, callus formation. Callus formation is the result of unlimited mitosis. In case of animal cells, the abnormal mitosis causes cancer, and in case of plant cells, the abnormal division causes for callus formation. The arrangement of homologous, that means same type and size of chromosome, that means homologous chromosomes. Homologous chromosomes are enter from maternal and paternal parts or body. The arrangement of homologous chromosomes of an individual in descending order according to size is known as karyotype. Karyotype. What is karyotype? It is the arrangement of homologous chromosome. It is the arrangement of homologous chromosomes of an individual. In descending order, arrangement in descending order, larger to shorter ones. Descending order according to size is known as karyotype. These homologous chromosomes are passing from parents. There are 23 pair of chromosomes, 22 pair of autosomes and 1 pair of sex chromosomes. In autosomal cells, the mitosis are occurred. Autosomal cells, that is body part except the reproductive organs. Now we complete this part. If you like this portion, please like, share and subscribe the channel.